Welcome to Educon. In this video, we will learn about cell envelope and its modification in prokaryotic cell. Most prokaryotic cells, particularly bacteria, they have a chemically complex cell envelope, and this cell envelope is tightly bound 3-layer structure made of glycocalyx, it is the outer layer, followed by cell wall and then plasma membrane, and each layer they perform distinct function, and all together they form a single protective unit. Single protective unit. So, in bacteria this cell envelope is a chemically complex, and also it is a tightly bound 3-layer structure which consists of outer layer glycocalyx followed by cell wall and their innermost layer plasma membrane. So, all together they form a single protective unit. In certain bacteria, the composition on the cell envelope differs, like in few bacteria it is like loose sheath where it is called as a slime layer, and in certain bacteria it is thick and tough where it is called as a capsule. So, the outer layer will be called in different ways based on the composition on the cell envelope and the glycocalyx, consider that fact. And the bacteria classified into two groups, okay, same based on the composition of the cell envelope and also in the manner how they involved in the gram staining procedure developed by a gram witch. So, if the bacteria they took the staining, then the bacteria are called gram positive. If the bacteria does not involve in staining, that does not take the stain, they are called gram negative bacteria. So, this gram witch is the scientist who developed the staining procedure, and the bacteria which take the staining they are called gram positive bacteria, and the bacteria which do not take the staining they are called gram negative bacteria. Coming to cell wall. Cell wall gives structural stability and prevent the cell from bristling and collapsing. So, it holds the cell, and we have the innermost layer called plasma membrane which is selectively permeable. Remember this, the plasma membrane is selectively permeable, and there is membranous structures called mesosomes which are formed by the infolding of the plasma membrane, and they are present in the form of vesicles, tubules and lamellae. So, these are present, the mesosomes may be present in the form of vesicles, tubules, lamellae, and mesosomes helps in cell wall formation and DNA replication and distribution to daughter cells, and secretion process, increasing the plasma membrane, increasing the area of the plasma membrane for enzymatic activity. So, and it also helps in cell respiration. In certain bacteria like cyanobacteria, they too have membranous structures like the infoldings of the plasma membrane, where they are called as a chromatophores which contains pigments in them. Bacteria can be motile or non-motile. By being motile, they have flagellum, which is useful, which helps the cell in movement, which is made of filament, hook and basal body. Flagellum. So, if the bacteria is motile, then they will be having the flagellum, which helps the bacteria to move, and it is a filamentous extension from the cell wall, and it is the longest. The flagellum, the filament is the longest part. It consists of 3 parts: filament, hook and basal body. And in certain bacteria, we have pili and fimbriae. Pili are tubular structures made up of special protein, whereas fimbriae, they are like bristle like. So, it helps the bacteria to stick onto rock on in any stream of water, also to host tissue. Ribosomes and inclusion bodies in prokaryotic cell. In prokaryotes, ribosomes are associated with the plasma membrane. And ribosomes also are site for protein synthesis. In prokaryotes, ribosome is made of 2 subunits. One is the larger subunit is called 50S, and the smaller one is 30S subunit. Together they form 70S prokaryotic ribosome, whereas in eukaryotic cell the ribosomes is of 80S. So, there the larger subunit will be called as 60S subunit and the smaller subunit will be 40S. So, what we discuss here, ribosomes are a site for protein synthesis. On the image here, you can see the mRNA, okay, on that ribosomes are present. So, ribosomes along with mRNA are called polysome or polyribosome. So, here this is how the protein synthesis occurs in prokaryotic cell. Ribosome translate mRNA into protein. Now, we will discuss about inclusion bodies. So, what are inclusion bodies? In bacteria, reserve materials are stored in the cytoplasm in the form of inclusion bodies. There is no membrane around the inclusion body. So, they are freely moving in the cytoplasm. No membrane bound organelles. Same for the ribosomes. Ribosomes are also, they are non-membrane bound organelles found in both prokaryotes and also eukaryotes. And the reserve materials is in the form of phosphate granules, glycogen granules, cyanophycean granules. So, what are the different types of examples for the phosphate granules, glycogen granules, cyanophycean granules? And gas vacuoles are found in blue, green, purple and photosynthetic bacteria. Gas vacuoles. These are also examples of inclusion bodies in different bacteria.